DNA and the genome. In this lesson, we'll look at the science and engineering practices of constructing explanations and designing solutions, as well as obtaining, evaluating, and communicating information. Our cross-cutting concepts will consist of patterns, cause and effect, scale, proportion, and quantity, and structure and function. So what is the genome? The genome is the term used to describe the entire genetic material of an organism. The genetic material of an organism is made from the chemical called DNA. DNA stands for deoxyribonucleic acid. DNA molecules carry the genetic code that controls the function of cells and the characteristics of an organism. Where is DNA found? DNA is located inside the nucleus of cells in the form of chromosomes. Each chromosome is made from one long molecule of DNA. A gene is a small section of DNA on a chromosome. Each gene provides the instructions to make a particular type of protein. Recall that human body cells contain 46 chromosomes in 23 pairs. So where are genes located? What is the body made up of? How and where is genetic information carried in the body? The body is made up of different types of tissues. These are made up of specialized cells. 2 or more tissues can come together to form organs. Skin is made of epithelial tissue. Cells are the building blocks of tissues. Each type of tissue is made up of a group of specialized cells. Specialized cells are adapted to carrying out a specific role. The nucleus is the control center of a cell. It contains the cell's genetic information. What name is given to the structures found inside a cell's nucleus? These are chromosomes. Chromosomes carry the genetic information located in the nuclei of cells. Each chromosome is made of a single molecule of DNA. A gene is a small section of DNA on a chromosome and controls a particular characteristic. There are hundreds of genes on each chromosome. Producing proteins. The genome contains the instructions to make all the proteins in an organism. Each gene produces instructions for a specific sequence of amino acids. These then join together to produce a particular type of protein. The gene instructs a specific sequence of amino acids to join, forming a protein. And that is read from a section of DNA. Proteins control the activity of cells. As a result, proteins are essential in determining how an organism grows, develops, and functions. So how do genes control characteristics? How do genes control the growth and development of whole organisms and their characteristics? Stage number 1. Each gene provides instructions that tell a cell how to make a particular protein. Proteins are the very important structure inside of the cell and allow the cell to function. They perform many roles within the body. Stage 2. Some proteins are structural and are used to form cells and tissues. Others, like enzymes, are involved in chemical reactions and other body processes. So how does this create a whole organism? Stage 3. Proteins, like enzymes, can produce other substrates or catalyze reactions. This results in the formation of a whole organism. Cellulose, which gives plants their structure, is produced by enzymes. Stage 4. The control of characteristics starts on a tiny molecular scale through the action of DNA and proteins. However, it results in whole organisms growing and developing, from trees to whales. Genotype and phenotype. The genome forms the genotype of an organism, the genetic information in the genome that contributes to the characteristics of the organism. The genome interacts with the environment to form an organism's phenotype, the set of characteristics that are observable in an organism. The phenotype depends on both genes and the environment. Most characteristics rely on instructions in the genome, but they are modified as a result of the interaction between the organism and its environment. Genes in the environment. How can we observe the interaction between the genotype and the environment? Imagine a pair of identical twins. They have exactly the same genotype, yet their observable characteristics are slightly different. This is because a person's phenotype is a result of the interaction between their genes and the environment. Some of the aspects of the environment that could interact with the genotype to influence phenotype include diet, exercise, smoking, stress, disease, piercings or tattoos, haircuts or makeup. All of these are examples where the environment may cause the phenotype to slightly differ. Passing on genetic information. In all living things, characteristics are passed on in the chromosomes that offspring inherit from their parents. Chromosomes are found in pairs inside the nucleus. In each pair, one chromosome comes from the father and the other from the mother. Each of the chromosomes in a pair contain different versions of the same genes. These are called alleles, and each version is called a variant. True or false? Are these statements about the genome true or false? Genotype is the term used to describe the proteins in an organism. That's false. The genotype builds the proteins. An organism's phenotype can be influenced by the environment. This is true. In a pair of chromosomes, one has been inherited from the father and the other from the mother. This is also true. The genotype of all organisms is made from DNA. This is also true. An allele is a version of an amino acid. That is false. The structure of DNA. DNA is a polymer. This means it is a large molecule made of many smaller units joined together. Each of these smaller units is called a nucleotide. Every molecule of DNA is made from 2 strands. Each of the 2 strands is made from thousands of nucleotides. DNA molecules form a special spiral shape that we call a double helix. All right, let's match some genetic terms. A chromosome is a long, tightly coiled molecule of DNA. DNA is a polymer made from the chain of nucleotides. The double helix is the shape of a 2-stranded DNA molecule. And a gene is a section of DNA that codes for a sequence of amino acids. Structure of nucleotides. DNA is made from 4 different types of nucleotides. Each nucleotide consists of 3 main parts: a phosphate group, a sugar, and a base. The base is the only part of the nucleotide that can vary. There are 4 different bases, each producing a different type of nucleotide. The 4 different bases are adenine, cytosine, guanine, and thymine. Typically, we shorten these to just A, C, G, and T. The bases of different nucleotides can interact with each other. The DNA backbone. When nucleotides join together, their sugar and phosphate sections form long strands. A single DNA molecule is made of 2 strands of nucleotides. This forms the backbone of a DNA molecule. The bases on each nucleotide face inward, towards each other. This allows bases on opposite strands to interact, keeping the 2 strands together. What is a complementary base pair? DNA molecules are double-stranded. The base of a nucleotide on one DNA strand can interact with the base of the nucleotide opposite it on the other strand of the DNA molecule. If this happens, the 2 nucleotides are said to have formed a complementary base pair. Only certain pairs of bases can interact to form complementary base pairs. Base A can interact to pair with base T. Base C can interact to pair with base G. And that is the only arrangement that can happen. As a reminder, A means adenine, T means thymine, C means cytosine, and G means guanine. Bonding between complementary bases. Weak hydrogen bonds form between complementary base pairs. The hydrogen bonds hold the 2 strands of nucleotides together and maintain the double helix shape of DNA. Recall, C always pairs with G, and A always pairs with T. Hydrogen bonds are not as strong as chemical bonds. This means they can be easily broken by heat. If DNA is exposed to heat, the hydrogen bonds between the strands break and the molecule loses its structure. So what is the structure of DNA? Let's start by unraveling a chromosome. A chromosome is all of the chromosomes are made from a single molecule of tightly coiled DNA. Genes. Each gene is a small section of DNA on a chromosome. Each gene contains instructions for a sequence of amino acids. These join to form a particular type of protein. The double helix. DNA double helix. The 2 strands of a double-stranded DNA molecule twist to form a special spiral shape called a double helix. DNA strands. A DNA molecule is made of 2 strands of nucleotides. We say that DNA is double-stranded. Nucleotides. Each DNA molecule is made of many nucleotides joined together. DNA is a polymer. Each nucleotide is made up of a phosphate group, a sugar, and a base. Order matters. The type of protein produced by a gene depends on the order of bases in that section of DNA. A sequence of 3 bases codes for a particular amino acid. This means that the order of bases controls the order in which amino acids are assembled. Every 3 bases code for 1 amino acid. The order of amino acids determines the type of protein produced and how it folds to form its specific shape. Shape is very important to the function of proteins. For example, The shape of an enzyme's active site determines what type of reaction it is capable or able to catalyze. DNA and protein synthesis. What is the link between genes and proteins? The genetic code controls cell activity by coding for the production of specific proteins. DNA and bases. DNA contains a long sequence of chemicals called bases. The sequence of the bases provides the genetic code for the production of proteins. Pairs and triplets. Bases on opposite strands of the DNA molecule link to form base pairs. 3 base pairs make up that triplet. Triplets and amino acids. Different triplets code for different amino acids. The order of triplets determines the order of amino acids. Each protein is made of a different sequence of amino acids. If we look at this image, we've got the first triplet coding for amino acid number 1. The next triplet coding for amino acid number 2. The third triplet codes for a different amino acid. And finally, the fourth triplet again is coding for a different amino acid. This 3-stage sequence introduces the link between genes and proteins before the mechanism of protein synthesis is explored in our next section. Mapping the human genome. The Human Genome Project was an international, publicly funded scientific research project. Its aim was to sequence the entire human genome and map the location of genes on the chromosomes. The project was completed in 2003 and revealed that the human genome is made up of around 3 billion bases, forming 25,000 genes. Information revealed by the Human Genome Project has enabled further discoveries relating to medicine, biotechnology, and human evolution. Medicine and the human genome. Access to the human genome sequence has led to research to try to link diseases to specific regions of the genetic code. The Human Genome Project has enabled the discovery of over 1,800 genes that are thought to be involved in disease. In the future, it is hoped some diseases could be diagnosed before symptoms appear. This could be done by analyzing a person's DNA sequence to see if they carry any genes identified as a risk for disease. Diseases that could be diagnosed before symptoms appear using genetic testing include Huntington's disease, Alzheimer's disease, and some types of cancer. Inherited disorders. The Human Genome Project has helped scientists to understand how disease that run in families are inherited. If a disease can be linked to a specific gene, then genetic tests can be carried out to see if a person has the disease. Genetic tests. Genetic tests can be used to determine if an individual has a family background of inherited disorders by determining whether they carry the faulty gene. The results from a genetic test can help an individual make decisions concerning their own future health as well as whether to have children. Targeted drugs. Our increased understanding of the human genome has helped to develop drugs that target genes to treat disease. Genes are involved in many diseases. Using drugs that target such genes is a very powerful technique as it allows medicines to act with great precision. Cancer therapy is one area benefiting from targeted drugs. Drugs have been designed that target genes involved in abnormal cell division, with the aim of stopping tumors from growing and spreading to other parts of the body in patients. Human evolution. The Human Genome Project has enabled scientists to study how humans have migrated across the world over our history. DNA samples have been taken from populations around the world. These have been used to research similarities and differences in DNA sequence between populations living in different countries. It is now possible to take a genetic test to trace where an individual's ancestors came from hundreds and even thousands of years ago.